The same is true when it comes to testing, whether it's chemicals or pharmaceuticals. Animals are not sufficiently predictive of humans with regards to toxicity and to safety. You can never make an animal that you're testing human relevant enough.
Product safety for pharmaceutical and chemical products is critical. We need to be able to trust that our medications, foods, cosmetics, and soaps aren't going to harm us or the environment.
The science of product testing and toxicology led to massive increases in product safety over the course of the 20th century.
Unfortunately, much of that testing was done on animals, which has been a controversial topic for decades.
In the 21st century, we may be able to advance past animal testing with the help of computer modeling and advanced cell-based methods.
Hi, I'm Charis.
And I'm Jesse, and we're the hosts of the Analytical Wavelength, a podcast brought to you by ACDLabs.
In this episode, we're going to be talking about the role of chemical predictions in toxicology and product testing.
First, we're going to be talking to Dr. Jared Bailey, Director of Science at Animal Free Research UK. He's going to explain the scientific argument against animal-based testing. So hello, today I have Jared Bailey here with me. He is the Director of Science for Animal Free Research UK, and he did his PhD in genetics at Newcastle University. Is that correct?
That is correct, yes. Newcastle in the far north of England.
Excellent. I wasn't aware. My geography of the UK is actually surprisingly bad. I should probably learn a little bit more about that at some point.
Well, it's Newcastle seems to be most famous for its beer, for Newcastle Brown Ale, which arguably is bigger in North America than it is in England these days. But yeah, it's famous for its beer and for a pretty bad football team. But it's a nice place to be.
Okay, cool. Glad to hear that. Okay, well, thank you so much for joining us today. Our first question, of course, is what is your favorite chemical?
I gave this a little bit of thought and kept coming up with chemicals that probably aren't particularly exciting or are many people's favorite chemicals. But I'm going to go for this one. One of my passions since I've been at school, which is quite a long time ago now, is skiing. I love the mountains and I just love skiing. So it's actually probably something as simple as paraffin wax. It's something you can put onto your skis and go out and just have a fabulous day in the mountains sliding around on the snow. So a pretty simple answer, but one that's brought me a lot of joy over the years.
Yes, that's a good one. That's a good one. Lots of applications for chemicals in our life. So I want to talk a little bit about your background and kind of how you came into the field of animal-free research. Can you tell us a little bit about how you ended up there?
Yes, of course. Well, my degree was in genetics. My PhD was in virus genetics, which of course is very topical at the minute with COVID. I then spent 7 years as a researcher researching premature birth in humans. Why do women have premature babies? And of course, this has lifelong consequences for millions of children and people every year. So it's a pretty serious issue. It was during that time that I realized that my own moral issues with animal experimentation, I didn't think it was right to cause other beings who happen not to be human pain and suffering for science. That was augmented by a scientific argument. I was realizing that people were using animals to study the same thing I was studying, using human tissue and human cell culture. We were getting different results that conflicted with one another. And I started to think maybe this isn't the best thing we could be doing scientifically either. And of course, if it's not the best thing we can be doing scientifically, there are human ethical issues. Because we can be doing better things, more human relevant things, more humane things. And if we're not delivering cures and treatments, understanding of human diseases and biology, then there's a human ethical angle too. Cut a very long story short, I spent one lunchtime in the lab writing to, I think, about 35 different organizations that campaigned on this issue to try and get a change in how science is done. And for the last 17 years, I've worked for a number of organizations, both in North America and in the UK and Europe as a whole. underpinning the scientific argument to this, asking scientific questions. Are animals good models for human beings, both in research and in the testing of chemicals for chemical safety and new drugs for efficacy and safety? Publishing lots of papers, book chapters, making the case to governments and inquiries, the FDA, the EPA, the European Commission, and many, many more. And that's how I got into this. And very briefly, currently with Animal Free Research UK, I started work with them in July last year, so not too long ago. And as well as continuing to make the scientific case to try and encourage a shift from animal science to human-specific science through public affairs programs, through political avenues too, we fund a lot of human-specific research. We've funded more than £10 million worth of science in the UK, more than 260 projects. We, I think the last year, for which figures are available, about £750,000 were giving to scientists to do human-specific animal-free research and to educate early career scientists that this is the future of science, and to excite them about that, and to get them into that way of thinking. And just to finish off, I firmly believe, as more and more scientists seem to, that we need to maintain a human perspective from start to finish, whether it's in research, disease research, basic research, or whether it's in testing of chemicals and new drugs for safety. There are too many species differences between one species and another. There are many differences in the same species between individuals to worry about, but we need, from start to finish, to maintain a human perspective to do the best science. And I've been convinced of that for a long time now.
So let's get right into that then, the meat of it. So what would you say is your argument on that? Because I think that there are many people who have opinions on the ethics of animal research just from the perspective of animal welfare, and people probably have made decisions or have thoughts on that. But I think that this idea that there's a scientific argument for not using animal models might be new to some people.
Yeah, I mean, it's obviously for the purposes of our discussion, I guess it's going to be more interesting for the listeners to hear about testing of chemicals and drugs. But it's important to remember all the way through that this also applies to research. Are we really understanding, for example, many different types of cancers and how cancers metastasize and spread? Are we really getting to the bottom of Alzheimer's disease and Parkinson's disease and many, many others? Never mind finding treatments and potentially cures for them. I think the answer is no, we're really, really not. We've been led down too many dead ends because of the use of animal models that are of poor human relevance. The reason for that poor human relevance comes down to genetics. We are genetically too different. We, of course, share many of our genes, but even when we share genes between rats, mice, dogs, monkeys, cats, humans, everything else, those genes are expressed differently. They're doing different jobs in different ways, and that really, really, really matters. Those gene expression differences have been linked to all sorts of biological processes and diseases. The same is true when it comes to testing, whether it's chemicals or pharmaceuticals. We need to know how a drug or a chemical is absorbed, distributed around the body, metabolized, excreted, and so on and so forth. And all of those things differ often significantly between different species, even between different individuals of the same species. So the point is that we know that animals are not sufficiently predictive of humans with regards to toxicity and to safety. We know why. It's because many of the drugs that are involved in those processes, absorption, distribution, metabolism, excretion, those genes are different, and so you can never make an animal that you're testing a new drug or a new chemical on human relevant enough. We really have to maintain a human perspective. So it's actually a relatively simple principle scientifically. And there are two things that come from that. One is, should we be using animals at all? Because they're really not terribly predictive. And the second thing is, if we don't use animals, what should we be doing instead? And we can talk about this. That there are some amazing scientific technologies that would have seemed like science fiction to me 20 years ago when I was in the lab, that are enabling us to do this research and this testing in a human context. and it's much, much more predictive for what is going to happen to an actual human being. And if you like, I can tell you about some of the evidence my own work generated to show that testing new drugs on animals is not adding statistical evidential weight to the likelihood of toxicity or safety, and what that means and where we should go from there.
Yeah, I definitely want to hear more about these alternatives. It's something that I imagine that the science has been moving very quickly between advances in the cell culturing methods and then also in silico methods has been changing a lot recently. So I'd love to hear kind of where things are at and how these compare to animal testing models.
Yeah, so what's happening? I think it's important to say from the outset that maybe 20 years ago or so, this was, I guess, a relatively niche point of view. There weren't too many scientists shouting about this and demanding change. That's really, really changed. And now this is a mainstream issue. It's something that's being discussed and debated, researched, written about in mainstream scientific journals and conferences. There is an appreciation of a need to move to more human-specific research and testing within the regulatory agencies, the Food and Drug Administration, the Environmental Protection Agency in Europe, too. The European Parliament recently made a resolution to phase out animal testing because there was a scientific need to do so. So what do we do instead? What is giving science and scientists the confidence that we can do this paradigm shift, if you like, and we can move to better science? And I think it largely pivots around two or three things. One is advanced cell and tissue cultures, so-called organoids, the culturing of mini human organs, if you like. They're the size roughly of like a full stop or a fly's eye. They're three-dimensional. They are composed of many different cell types. In other words, they have real structural and functional physiological relevance to a real human organ. There's organ on a chip and body on a chip technology. So for those of your listeners who might not know what these are, they're very small chips. They're made of glass or silicon. They used to be the size of a smartphone. They're even smaller now in many instances. And they have these advanced three-dimensional cell and tissue cultures, human-specific, with circulatory systems that simulate blood flow. If you're looking at lung tissue, they have airflow that mimic breathing. If you're looking at kind of mini heart cultures, they can physically beat. They're really, really mimicking what human organs, tissues are doing and how they interact with one another. You can factor in the liver and the metabolism of new drugs and chemicals. So this is really, really exciting, and they're proving their worth in many areas of research. For example, neurodegenerative diseases like Alzheimer's and Parkinson's that I've already mentioned, and also in the testing of new drugs and chemicals. So there have been a couple of papers recently that have really demonstrated the superiority of these methods. One of the biggest issues is drug-induced liver injury or DILI. There's chemicals and including pharmaceuticals often. If there's an issue, they damage the liver. That's where they go. That's where they're metabolized, and the metabolites damage the liver. Now, that traditionally hasn't been predicted well by the use of animal tests. And experiments have shown that these liver-on-a-chip technologies are vastly superior to animal methods. And I think there was a paper published recently by a company called Emulate that were one of the pioneers in these organ on a chip and body on a chip technologies that showed their chips could predict liver toxicity with 87% sensitivity and 100% specificity. This is just on another level compared to what any batch of animal tests can do. And finally, this is now feeding into artificial intelligence too. There are AI companies that are getting tens of millions of dollars of investment, venture capital and investment, because their technology is so promising. So a lot of these data can feed into AI, and the computers, allied with computers, they can predict even better what real human beings are going to do, how they're going to respond to this particular chemical or this particular drug. And one final point, these kinds of technologies have also been used in repurposing of existing drugs for the battle against COVID with great success. So it seems clearer than ever, objectively and amongst the scientific community and increasing numbers, huge numbers of scientists, that this is the future of science and that there's a real need for the benefit of humans, not just the 200 million animals a year used around the world in science. There's a real need for this shift to a more human focus, and everyone's going to benefit from that.
Yes, it sounds that way. So with that, like, I think that you kind of were touching specifically on AI as one of these frontiers of animal-free testing, and I'd be interested to hear what else is there on the near horizon of either what are the challenges that you think might still be out there that need to be conquered, or what are the technologies that are maybe not quite ready yet for prime time, but you're excited about to see how it performs.
Yeah, I think it's important to say that we're really a lot further on than many people have previously thought. We're already at a point where we can do much better than animal-based approaches. Another critical point is that we can constantly improve human cell and tissue cultures, organoids, organ and body on a chip technologies, constantly improve computational methods. That's not something you can do with animals. Animals are animals. You can only genetically modify them to be a little bit more human-like to a certain degree. And there's always a huge ethical issue with that. So we're already in a better place. We're already in a place where these technologies that can replace animals are hugely advanced and can be improved even further. And there's another thing that feeds into a shift towards human-specific technologies, which is that you can factor in human variation and diversity. And you can do that by taking samples of blood or skin, for example, possibly even urine from people and harvesting cells from those samples, coaxing those cells to go back in time to something called stem cells. And if any of your listeners might not know what they are, when we were developing embryos, we were composed of cells that could turn into any of the specialized cells in our body. Lots of the different specialized cells could stem from those cells. You can coax developed cells in adult humans to think that they're stem cells again, and then coax them to be any type of cell that you want to investigate. Now that's important because if you can take cells from healthy individuals, varied healthy individuals, particular patients with particular diseases with specific different kinds of genetic causes, then you can model all of those different human individuals using the technologies I've spoken about. So not only are you being human specific, you're factoring in human variation and specific genetic causes of human diseases that you can compare directly with people who don't have those diseases. So it's a tremendously exciting time, and I think this is why when I speak to early career scientists, young undergraduates, they are arguably more excited about a career in science than I've ever seen in the last few decades, and so they should be.
Yeah, that sounds also particularly interesting, but the implications are something like personalized medicine, of being able to do specific testing on this and see how it would fare for a particular individual. What about resources for people who want to learn more about this? Because this is obviously, we're doing a survey of the field here. I'm sure that you could talk more about any one of these technologies or any one of these points. So what resources would you recommend for people who want to go deeper into the science of these questions?
Yeah, I guess it stands to reason what I'm going to say. Social media is a huge goldmine of information. Many of these biotech companies who are developing these technologies, many scientists and institutions who are using these technologies and generating amazing data. So get on social media, get on their websites and have a look. Have a look at our website, animalfreeresearchuk.org. See what sort of science we're funding. See what research that we're pointing towards that we find is amazing. Have a look on scientific repositories like PubMed. Do PubMed searches. Find out where organoids and organ-on-a-chip technologies are being used, what sort of data they're generating, and how excited scientists are about the data that are coming out of these technologies. So there's all sorts of resources, largely internet-based, but you could spend years looking through the information already, and it's tremendously exciting.
And I'm sure that there are people who do.
Yes.
So to spend lots of their time looking through, but that'll be great for this as an introduction to that. Thank you so much for your time, and it was lovely chatting with you.
It was a pleasure. Thank you for the opportunity.
Jared offered a fascinating take on the scientific challenges with animal testing.
And it isn't a fringe view. In fact, companies like Unilever have made it clear that they feel animal testing is unnecessary.
While not everyone may recognize the name Unilever, you'll certainly recognize some of their brands. Dove soap, Ben & Jerry's ice cream, and Axe body spray, to name a few.
Dr. Steve Gutzel is a team leader and computational scientist at Unilever. We talked to him about the role of computational chemistry in product testing.
Here with us today is Steve Gutzel, who is a computational science and team leader at Unilever in the UK. Steve worked for 18 years with Unilever, and he received his PhD in organic chemistry from Swansea University. Hello, Steve.
Hi, nice to talk to you today.
It's great to have you. So let's start today with our usual icebreaker question. What is your favorite chemical?
Okay, yeah, I gave this one some thought, and it's a little bit cheesy, it's a little bit corporate, but forgive me. So there's a class of surfactants, the alkylocethionates. I've spent a long time in my career working on surfactants, studying their properties. They're a tricky bunch of chemicals, but we've got some really cool models now to predict how these molecules interact with biological membranes and cause toxicity. So the science has really moved on even within my career. So those alkylocethionates are a series of chemicals that I've kind of looked at and studied throughout my career. And they just happen to be the active ingredient in one of our leading products, the Dove Bar. It's probably one of our oldest products, but you know, if it's not broken, don't fix it. It's a great product. So yeah, alkylocethionates for me have been good fun over the years.
That's really interesting to hear, and I imagine that even this idea of picking compounds just because they're new versus whether going to things that are old and still work is probably something that comes up in your work a lot. But let's get into that. Can you tell us a little bit about how you got into computational toxicology?
Sure. So my PhD, as you mentioned, in Swansea, it was actually organic chemistry. So I was based in the lab. So it was actually part funded by Unilever, and it was linked to ecology. So even from my postgraduate studies, I was studying how can you link the properties of chemicals to some kind of toxic effect. In this case, it was skin sensitization. Will a chemical result in an allergic reaction in people? And the fascinating thing to me that kind of crystallized during my PhD was that all of that information is wrapped up in the chemical somehow, in the chemical structure. So if you understand enough of the chemistry, and critically, if you understand the biology, the toxicology side as well, you can actually start to relate the two things together. So what I did was I was measuring stuff in the lab. I was measuring kinetics, rate constants, so small organic molecules reacting with model nucleophiles, so a model of a protein side chain. That's the fundamental step in causing skin allergy. And what I found out was that, yeah, you can fairly easily measure these things in the lab, and it can inform you about the biological properties. So not only could you predict whether something would cause dermatitis, allergy, or not, the really cool thing was by understanding the magnitude, in this case, the rate constants, you could do quantitative predictions. And the link to Unilever really came into its own. I came and did placements at the lab where I am now in southeast of the UK, and the guy that I worked with was a computational chemist. So like all computational chemists, there's a little streak of arrogancy there, and he was like, well, you're measuring all this stuff in the lab, that's really difficult. Surely we can do this in the computer. So he taught me how to do some really cool quantum chemical calculations, some semi-empirical calculations, molecular orbital energies, effectively recreating what I'd done in the lab in the computer, which had I known that 3 years earlier, maybe my PhD would have taken a slightly different course. But that was the hook for me then, working out that not only could you measure these things in the lab and relate them, but you could then recreate those experiments effectively in the computer and build these predictive models. And then it was a kind of bit of serendipity, right place, right time. That particular scientist moved on from Unilever, creating a vacancy. And I thought, well, why not? Why not me? Why not put my CV in? And I remember the hiring manager saying, well, yeah, this is a computational position. You're not really a computational chemist, but we think you could be. We've invested in you and we want to continue that. So yeah, it was a combination of things, but that real insight gained through my PhD, I think, is what hooked me in.
That's great. Going into the computational side, from my understanding is a core part of that is quantitative structure-activity relationships, or Qsar. Can you give us a bit of an introduction of how Qsar works in predictive toxicology?
Sure. As the name suggests, what you're trying to do is relate the structure of a molecule to some kind of activity. And in our case, that's biological activity, and it's something related to toxicology. So really what you need to build a Qsar is three things. You need a set of chemicals with measurements of that property that you want to predict. So let's use the skin allergy as an example, as I already mentioned it. So what you would need is a set of chemicals that you already know the answer for. You already know whether they're going to be a skin allergen or not. But it could equally be a different property. It could be physical chemical properties. It could be environmental properties. Anything that you want to predict, something that's already been measured. The second thing that you need is a way to describe your chemicals. And you need to be able to do that in numbers, because what you're trying to do is build a predictive model within a computer, and computers, for all their benefits, they don't understand chemistry. So you have to introduce the chemicals into a model using numbers. So what we do is we basically translate chemical structures into descriptors. Now, they could be from the lab. So I mentioned in my previous work, I measured things in the lab. I measured rate constants. An experimental rate constant is a very good descriptor of a chemical in terms of how quickly it reacts with something. But of course, you can calculate these things. And the advantage with the calculations, you can calculate many, many more of these. And you can even calculate for chemicals that you don't physically have in the lab. So now it's possible to calculate thousands of descriptors on thousands of chemicals at the touch of a button. So they're your two bits of information that effectively allow you to produce a data matrix. So you have a property that you want to predict and a whole bunch of numbers describing the chemicals. And then you need some maths. You need a statistical method to go look for patterns. Because what you're trying to do is show that there's a relationship between some of those properties of the chemical that you've either measured or calculated and the property that you want to predict. So do chemical kinetics relate to skin allergy in my case? And the general rule is with the mathematics to keep it as simple as possible. If you can draw a straight line between the points, you're in a really good place. That's often not the case, and you do have to go to more and more sophisticated methods. But Qsars can start from literally a simple linear regression to show that your X variable is linearly related to your Y variable. So as reactivity increases, allergy increases, as an example. Or they can be extremely complicated using lots of descriptors and something like neural networks or random forests, more machine learning, AI techniques. And it's important to consider really when you're using these things, what are you using them for? Because it may be perfectly acceptable to have a very complicated model with really complicated AI, but if you have to explain the predictions and you need some transparency in that model, that may not be the best route. So for example, if you're predicting toxicity and you're trying to show that you don't need to do a traditional animal test, for example, those models may not be the best ones. It may be better to scale back and use something far more simple that you can actually explain, talk through, rationalize. But if you're in the business of coming up with a new lead drug, for example, and you're going to screen tens of thousands of structures, using more of a black box model that's very, very quick is absolutely acceptable because you're going to follow that up with subsequent testing.
Fascinating. There's a lot to it. But I'm actually really curious about kind of like what this looks like practically. From what you're describing, it definitely doesn't sound like people just, walk up to a computer and throw out a structure, press enter, and spit out an answer. Let's say that somebody comes to you with something like this, like surfactant molecules, like a new version or something like this. Like, what does that kind of look like practically for either you or your team to then turn that into this passes or this fails or these are the problems you should be concerned about?
So first thing, especially with surfactants, is to characterize the heck out of it. Because as I mentioned, surfactants, particularly commercial grade surfactants, are quite complex mixtures. So they're complicated chemicals and then they're complicated mixtures of individual chemicals. And it really is important to understand that because, for example, if you take the chain length of a surfactant, it can vary from, say, a C8 to a C18 or 20. The properties of those two different ends of the spectrum are very, very different. So understanding the relative proportions of those chain lengths, for example, will be critical to understanding the properties of that particular surfactant. So that's the first thing, is to really understand what it is that you're dealing with, and also accepting that there's a bit of variability involved as well. So it's, again, the nature of commercial grade substances versus kind of pure lab grade samples. So understanding what the variability will be over the lifetime of the product. Then we would look at its fundamental physical chemical properties. So things like its partition coefficients. Something really simple like the octanol water partition coefficient is a really good surrogate for lots of things in biology. Partitioning into membranes, between environmental compartments, those kind of things. For surfactants particularly, octanol water is not the best system. Surfactants by their nature like to sit on surfaces or interfaces between things, and they will quickly turn a practical octanol water experiment into a foaming mess. So some of the cool models I mentioned at the start, it's now possible to measure specifically partitioning between water and a biological membrane. You can do this on HPLC columns. It's really neat stuff. You can also do it in the computer. You can model a phospholipid bilayer and the interaction of surfactants with that. And that interaction with membranes is a critical determinant in how the toxicity of surfactants manifests itself in the environment. Surfactants are produced in really large amounts. Generally, during use, they end up going down the drain. And whilst the vast majority of what goes down the drain biodegrades, so effectively gets digested by bugs and even anaerobic degradation, some may end up into the aquatic environment, and you need to understand whether that's going to have an impact. Understanding how things interact with membranes in aquatic organisms is the fundamental mechanism behind the Qsars. And actually, these membrane water partitioning models are a much better surrogate than the octanol water partition coefficients. So another thing that we would investigate is something called the molecular initiating event. This is a concept of the first thing that a chemical does when it interacts with a biological system that could lead to some toxicology. So for my skin allergy example, it's chemical reactivity. Could it react with a protein side chain? But also you'd look at, could it bind to receptors or interact with enzymes? And these types of models are all very tractable from a computational chemistry point of view. So we've worked closely actually with some academic groups, Cambridge University in the UK, to build a suite. We now have models for over 100 protein receptors, and we can screen new chemicals against those. And it gives an early indication of this chemical could bind to this particular receptor, which could be linked to these particular toxicological outcomes. So characterize it, study the physical chemical properties and the impacts those could have, and then into things directly related to toxicity, so molecular initiating events or direct predictions of toxic effects.
Mm-hmm. You touched upon this briefly when you're talking about the aquatic toxicity and environmental effects, but we were interested in the role that computational chemistry plays in areas other than what people would normally think about, which I think is human toxicity. So are there other areas, and what is the role played there?
Yeah, so environmental safety, there's a big role for computational chemistry in there as well, both on the exposure side, but also on the effects side. So if you think about risk in terms of toxicity or ecotoxicity, it's a function of two things. It's a function of the hazard. So can the chemical cause a nasty effect, whether that's in humans, an allergy or whatever it happens to be, or in an aquatic species, it tends to be more crude measures of impact. And the other part of the equation is the exposure. How much of the chemical will the organism in the environment actually be exposed to? And the properties of the chemical impact on both of those. So the physical chemical properties impact where a chemical will end up in the environment. Will it partition to the air, stay in the water? Could it biodegrade? All of these things have an impact. And in terms of the effects, the hazards, Qsars for predicting toxicity to aquatic species were actually some of the first to be developed. They've got a history of around 100 years, and the US EPA in particular have been big advocates of the use of QSAR to predict, for example, fish toxicity. So a huge role for computational chemistry in the environment.
Now, regulatory requirements obviously play a really big role in how all of this works, how toxicology is studied and assessed. How have you seen changes on the regulatory side affect your work throughout the time that you've been working at Unilever? You've been there for now 18 years, so I imagine there's been some regulatory changes over that time.
Yeah, absolutely. So some regimes are very progressive and lead the way. So people like the US EPA, Health Canada, Environment and Climate Change Canada are very early adopters of computational methods, and indeed they're developers of these methods. A lot of the Qsars for fish toxicity that I've mentioned originated from the US EPA. Other regimes are willing to talk, and they are willing to change. So we, as Unilever, we actively go out and advocate for these changes, moving away from animal testing to alternatives. And if you take somewhere like China, where the regulations can be quite strict, the Chinese regulators are actually willing to talk to industry, to learn and to adapt, which is really refreshing. And then at the other end of the spectrum, there are some regulators who are a little bit stuck in the dark ages. There have been changes, but it's really variable depending on the geography and the sector.
Yeah, so you touched upon Unilever's advocacy in the area of moving away from animal testing. Can you explain a bit more what your company's position is on that?
Yes, it's really simple. We believe that there are alternative, better ways of ensuring that people and the environment are safe from the use of chemicals without using lots and lots of animal tests. The science has moved on a huge amount, so not just the chemistry and the computational methods, but the in vitro world, the systems biology, the machine learning. It's all so much further on than it was when I started, and it continues to evolve. And this is why, as Unilever, we go out and we share our science, we publish, and we present our results all of the time. We also collaborate with lots of different groups around the world to try to advance the science. So that's essentially wherever the best science is happening. So academics, industry, NGOs, and regulators. In the past, we've seen that regulations traditionally have lagged behind the latest science. So scientific developments may be that you can do something in a new way, and it takes a number of years for the regulations to catch up. But we feel that trend has got to stop. And together with our collaborators, we're really on a mission to make that happen. So rather than just complying with requests for new animal test data, we're pushing back, and we're pushing the alternatives to force the discussion directly with the regulators where we believe that is scientifically the right thing to do.
Well, it's very admirable for you folks to be pushing for that, and I'm very interested to hear how those affect things over time. But thank you so much for your time with us today, Steve. It's been lovely chatting with you.
Excellent. Thanks very much for the invitation. It was great talking with you.
Yes, thank you.
Great talking.
It's exciting to hear about all the progress in this field. These tools are only getting stronger, which means better predictions.
If you want to learn more about computational chemistry software, check out the Predictive platform from ACDLabs. We'll leave a link in our show notes.
That's all for this episode. In our next episode, we'll hear the case for CASE, computer-assisted structure elucidation.
Remember to subscribe to the Analytical Wavelength so you never miss an episode.
The Analytical Wavelength is brought to you by ACDLabs. We create software to help scientists make the most of their analytical data by predicting molecular properties and by organizing and analyzing their experimental results. To learn more, please visit us at www.acdlabs.com.